So a stem cell essentially is an undifferentiated cell that can differentiate into lots of different types of cells. So if you remember our cell specialization video, which you can check out if you haven't seen, undifferentiated cells tend to develop over time into differentiated cells. So for example, we had nerve cells or different types of cells in plants, xylem cells, phloem cells. Those are all examples of differentiated cells. But at the beginning, you have these undifferentiated cells. And with humans, you have what we call embryonic stem cells. And as you might have guessed, these are located in the embryo of humans. So when the sperm cell first fertilizes the egg cell, you produce these embryonic stem cells. And the idea is these can then develop and differentiate into any kind of cell that the human needs. And obviously that's really important when you think about the growth of a fetus. It needs all the different types of cells. It needs nerve cells, muscle cells. So it needs a starting embryonic stem cell that can develop into all of these, okay? So that's embryonic stem cells. In an adult human, you do have what we call adult stem cells as well, which work in a similar way, but they're not as effective as embryonic stem cells. So they can develop into certain other types of cells, okay? So for a good example of this is the stem cells that you find in bone marrow. So bone marrow is basically the substance that's on the inside of your bones. If you crack your bone open, you'll see bone marrow. Same thing if you crack like chicken bones open, you might have seen the inside is what we call bone marrow. And these can essentially differentiate into a few different types of cells. So the cells in bone marrow, for example, can differentiate into different types of blood cells. So you could produce white blood cells or red blood cells, for example. So this is useful. It's still an example of a stem cell that can differentiate into more specialized cells. But the idea is that it's more limited. This bone marrow cell, the stem cells you find in there, wouldn't be able to differentiate into muscle cells or nerve cells or anything that you need. But the embryonic stem cells are much more effective and they can differentiate pretty much into anything you want. We can also look at an example in plants. So with plants, you have what we call meristem cells or meristem tissue. And these are present throughout the lifetime of a plant and it can differentiate into any type of plant cell. So if you remember the different types of plant cells like we saw, for example, the xylem cell or the phloem cell, or the root hair cells, when you have meristem tissue in the plants, these are types of stem cells that can differentiate into any of those plant cells that you need. So you kind of referenced there that stem cells are pretty useful. And if we take the example with a plant, so if you have meristem cells, this can be really useful for farmers because what you can do is you can use those stem cells from the meristem tissue to produce clones of plants quickly and economically. So it's quick and it's pretty cheap because all you do is you take a cutting from a plant, you take the meristem cells from that, and therefore you can then take those cells, and those cells can be put anywhere and it will grow into that same plant. It's essentially a genetic clone of that plant. And this is really useful because you can clone rare species. So if a plant is on the verge of extinction, you can clone them to protect them from extinction. And you can also, if you think about growing crops, you can grow crop plants which are disease resistant. So the idea is genetically some plants might be resistant to diseases more than others. So you can clone those types of plants to grow a whole crop of those disease resistant plants. So this is really useful for farmers. Okay, so that's one application of stem cells in plants. An application which is slightly more controversial is the use of stem cells in humans. So what we call therapeutic cloning. And essentially, let's say you're suffering from paralysis or something like diabetes, where some of your cells are not working. So for example, in paralysis, maybe you're missing some nerve cells. What you do is you take a stem cell and you grow it into a new nerve cell. And then you place those new nerve cells into the body of the paralyzed person. And therefore, they can then start to feel because now they've got those missing nerve cells. Okay, so that's one example of the use of therapeutic cloning. But in order to get those stem cells, well, we just looked at it before. Where do you get it? You get it from the embryo. Because remember, embryonic stem cells are the more powerful ones. They can differentiate into anything. So if you want to grow nerve cells, you need to go and get an embryo from the patient. And so you would essentially artificially fertilize an egg and a sperm with the genetic material of the patient, and they would grow those nerve cells that would then be compatible with the patient's body. And because the embryo contains genetic material from the patient, then when you transplant those new stem cells, then the body is not going to reject it. So a common theme when you do transplants is basically worrying about the patient's body rejecting the donor's organ or whatever they've donated. In this case, because the stem cell itself is from the patient, that means the new cells that you grow will also be from the patient, essentially. Will have the patient's genetic material, so the body won't reject it. Okay, but there are some risks of this. Essentially, if the patient has genetic defects, those will also be transferred. Okay, so one of the downsides, you can say, it transfers things like viral infections, which would be found in the DNA. The stem cell would also be grown with those same viral infections. A lot of people have ethical or religious objections. And the idea behind that essentially is the use of this embryo here. Essentially, you are fertilizing an egg cell for the purpose of not growing it into a actual baby, into a fetus, and then having a baby being born, but you're just using those cells in a way that essentially wasn't designed for. And therefore, some people have religious or ethical obligations to that, as they believe that's wrong, and that the embryo itself shouldn't be used as some kind of experimentation tool. Okay, so let's go into some questions to recap. Okay, a few questions as usual. Feel free to pause the video, have a go yourself, and come back for the answers. So question 1, what are the 2 types of stem cells humans might have? Well, you have your adult stem cells, and you also have your embryonic stem cells. And obviously the embryonic ones are only found in the embryo, so at the beginning of human life. Number 2, what is the stem cell tissue in plants called? So this is called meristem tissue. So hopefully you got that. Number 3, what are the advantages of therapeutic cloning versus transplanting cells from a donor? And the main advantage of therapeutic cloning versus just simply doing a transplant is that you reduce the chance of rejection. So remember, a lot of the time, the body of the receiver will actually reject the donor cell or donor organ. And in the case of therapeutic cloning, because it's the same genetic material as the person's body itself, there's much less chance of that rejection happening. Okay, so hopefully you got those ones correct, and I'll see you in the next one.